Andreas Pfenning. I'm here to talk about an NIH grant my lab just received to study the genetics and epigenetics of substance use disorders from NIDA, the National Institute of Drug Abuse. So the Avenir Award is given to new investigators. So it's a $300,000 in direct costs a year for 5 years, and it will kind of provide my lab a foundation to study the genetics and epigenetics of addiction in years to come. So it's estimated that there are over 2 million people in the United States with an opioid use disorder alone. And addiction broadly is a huge financial burden, but even beyond the financial cost, there's a loss of lives and loss of quality of life that's really hard to quantify. We actually know a lot about how addiction works. So essentially, these drugs of abuse hijack the reward pathway in the brain, leading to aberrant neural activity, leading to differences in neural circuits that ultimately impact the decisions that we make. So in parallel to all that we know about addiction at the neural circuit level, we're also beginning to learn more about addiction at the genetic level. So in particular, there are more and more genome-wide association studies that are coming out. And these genome-wide associations studies are telling us which parts of the genome are associated with addictive behaviors. A person is prescribed opioids for a surgery or some kind of chronic pain. And then after a certain amount of time on these drugs, that person becomes hooked. Now, another person might go in for the same surgery, might receive the same amount of opioid, but at the end of the process and at the end of their drug regimen, they don't have nearly as strong of an addictive drive to go back to opioids. And so a lot of that difference is mediated by a genetic component, right? So there are essentially different mutations in one genome compared to another genome that might make you more or less likely to become addicted to a substance like opioid or nicotine. We're beginning to gain a greater understanding of the genetics underlying opioid addiction and other types of addictive behaviors. So through genome-wide association studies, there are now literally tens to hundreds of locations in the genome that have been confidently linked to the behavior itself, which means that one mutations or set of mutations has a known influence on the addiction behavior. But where I think we know a lot less is we still don't know how those exact regions in the genome and how those mutations end up impacting the neural circuit side of things. So where my research comes in is trying to connect the two. So how do we go from a mutation or a location in the genome to the neural circuits that we know are underlying addiction itself? My group does a combination of computational research and experimental research, and we're especially interested in bridging the gap between the two. So on the computational side, we're developing and applying machine learning and artificial intelligence algorithms. And essentially what these do is make predictions about the system. So if we start with a segment of the genome that's known to be or suspected to be associated with addiction behaviors, we want to make a prediction about how that segment of the genome impacts the cell types and neural circuits that we already know are involved in the process. So we're also doing research on the experimental side. So we take the predictions that the computational model makes and we design experiments surrounding them. So for example, we developed a system where we can take segments of human genomic DNA and put them into the mouse brain and study how those segments function in the context of the neural circuits that we know are underlying addiction. So that allows us to take the static snapshot, so what the human genetic variants are, and study their behavior in the dynamic process of addiction itself. So it lets us measure the function of an addiction-associated segment of the genome when it's first responding to cocaine in the mouse, when the mouse is becoming addicted to cocaine, or in the future when the mouse is perhaps relapsing or being re-exposed to cocaine after a break. Studying the genetics of addiction is a valuable step in the process towards developing treatments for substance use disorders. Essentially, the work that I'm describing and more broadly, an understanding of the genetics can give us hints about which cell types are most relevant for the disease and also provide some insights into which biological pathways are going to be mediating the disease process. And once you know those cell types and the biological pathways within those cell types, you can start to develop drugs and treatments that target those mechanisms. You can find out more about this project by going to Twitter. You can find me at A Pfenning, A-P-F-E-N-N-I-N-G. And on Twitter, I have a link to the Avenir Award website where you can find out more about my research and the research of the other Avenir Award winners.